Hey everybody, it's Dr. Gilchrist here for Biopsychology. So for the next couple of lectures, I really want to focus on making sure that you understand action potentials. So I'm going to screencast a few of our lectures on drugs and neurotransmitters so that you have a better idea of these things and you can go over them in your book and you can go over these videos over the next week. Additionally, one of your essay questions will actually focus upon drugs, so I do want to spend some time talking about that so that you have a better understanding of it. So let's briefly define what drugs are. They are chemicals that affect the body. And in particular, we're going to be focusing on psychoactive drugs. These directly affect the central nervous system, the brain, and the spinal cord. And as such, they affect our things like our mood, our mental processes, and as such, they affect our behavior. If a drug is not a psychoactive drug, it's typically not large enough to cross the blood-brain barrier. Or in certain cases, it binds to proteins or ligands that make it too large to cross the blood-brain barrier. Now, for your essay question, one of them, you need to be able to understand what an agonist is, and you need to be able to understand what an antagonist is. In general, an agonist is anything that facilitates or enhances the effect of a neurotransmitter on the postsynaptic cell. On the other hand, an antagonist is anything that inhibits the effect of a neurotransmitter on the postsynaptic cell. So to help you kind of understand, and this is an incredibly complicated figure, I'll try to spend a little bit of time on this, but I would also recommend you check this in your book. This is really going to help you answer one of our essay questions. So one of the things that we know about drugs get the power that they do by working on different neurotransmitters. Drugs don't work by magic. They affect neurotransmitters, and as such, that's where they get their power. So there are many different ways that drugs can either serve as an agonist or as an antagonist. So you can kind of see that in this figure, agonists are kind of coded in purple, and then the antagonists are kind of coded in peach. So I'm going to start by talking about some of the different ways that a drug can actually serve as an agonist first, and then I'll talk about the antagonistic functions. So one of the things that we know that can enhance facilitation of a neurotransmitter is if a drug actually serves as a precursor. So some neurotransmitters are too big to cross the blood-brain barrier on their own. So if somebody has Parkinson's, we can't really just give them a pill of dopamine because it's not big enough to cross. What we can do, though, is provide a precursor that will make its way into the cell, and that precursor can be used to produce more neurotransmitters. As such, something like L-DOPA is actually an agonist for dopamine because it's a precursor. So it enhances that release. One of the other things that we can find is that certain types of drugs will actually stimulate the release of neurotransmitters. So in the case of black widow spider venom, it contains a chemical called latrotoxin that actually causes release of acetylcholine from the vesicles and from the cell. So acetylcholine floods into the synapse and causes muscle contraction. So if black widow spider venom is in your system and it does not get treated, you will develop what is called a rigid paralysis. Your muscles will contract and you can't relax. One of the other things that drugs can do is that they can actually stimulate postsynaptic receptors. So things like nicotine, which you find in tobacco products, actually enhance the effectiveness of acetylcholine at that receptor. And as such, it's an agonist. We have on the presynaptic cell something that's called an autoreceptor. Now, you won't need to know this for the test. Autoreceptors basically keep track of how much neurotransmitter is being released. And they can, if they're stimulated, they can actually cut production of that neurotransmitter so that less gets released. If we block that autoreceptor, that will increase the release of neurotransmitter. As such, it's an agonist. One of the other things that we find is that after a neurotransmitter has made contact with a postsynaptic cell, it can actually be taken back up into the cell and be recycled. This is a process that's known as reuptake. If reuptake is prevented, the neurotransmitters can stay in the system for longer and enhance the effect of the neurotransmitter. So a lot of antidepressants actually work this way. And in this case, that will be an agonistic function. Blocking reuptake means that the neurotransmitter effects are enhanced. One of the other ways that we can break down a neurotransmitter is basically prevent enzymes from breaking down those neurotransmitters. And again, that would be an agonistic function. Now let's talk about some of the antagonistic functions. So remember that an antagonist is anything that inhibits neurotransmitter production or release. So we have drugs like reserpine, which we'll talk about later, that prevent storage of neurotransmitters in vesicles. If they can't get stored, they can't get released. As such, it's an antagonist. A drug can inhibit the release of a neurotransmitter. So something like Botox will actually prevent acetylcholine from being released. And in this case, that will cause a flaccid paralysis. The muscles can't contract. Drugs can block postsynaptic receptors, thus not allowing the neurotransmitter to bind. Drugs can inactivate enzymes that are critical for production. And they can also stimulate autoreceptors. So again, if we stimulate an autoreceptor, that allows us to cut neurotransmitter production and release. So those are the different ways that drugs can serve as agonists or antagonists. I'd highly encourage you to look at the ones that you particularly understand and be prepared to know those because they are going to be on an essay question. So let's talk a little bit about the properties of drugs. Drugs do not work by magic. They mediate their effects through neurotransmitters. Now, some drugs like SSRIs, which are very common antidepressants, are selective and only work on serotonin. Not all drugs are selective, so they're not always selective for specific neurotransmitter systems. Some will enhance all neurotransmitters or inhibit all neurotransmitters. They can either be agonists or antagonists, and they have a lot of different sites of action, as we kind of saw in the previous figure. They can work on postsynaptic receptors. They can serve as precursors. They may affect enzymes that break down neurotransmitters, and they can affect storage or release of neurotransmitters. So we're going to move on to talk about the very first known neurotransmitter. It was identified in 1921 by Otto Loewy, who we will talk about very soon. His experiments showed that chemicals were the method of communication between neurons, and that's what those neurotransmitters are. Specifically, his experiment showed that if you stimulate the vagus nerve, which we talked about before, it's part of the parasympathetic nervous system, and it slows heart rate. Stimulating the vagus nerve causes contraction of the heart to slow, and the liquid containing the neurotransmitter, when transferred to another heart, would cause that heart to beat more slowly as well. Because the neurotransmitter was released by the vagus nerve, he called it Vagusstoff, vagus stuff or vagus substance, but we call it acetylcholine or ACH. It is widely distributed throughout the nervous system, both in the central nervous system and the peripheral nervous system. It's largely excitatory. In the peripheral nervous system, it facilitates muscle contraction. And additionally, it is the primary parasympathetic nervous system transmitter. Now, I am going to show you the chemical structure of acetylcholine, but this is not an OChem class. I will not ask you to draw this on an exam, so that's a good thing. Okay. So next, when we talk about a neurotransmitter system, we usually will add the suffix ergic, as you can kind of see here, to the name of the neurotransmitter. So neurons that produce acetylcholine are called cholinergic cells, and the pathways of acetylcholine action are called cholinergic pathways. There are 3 main acetylcholine pathways in the central nervous system. The nuclei of these cells are located in 3 major parts of the brain. Now, most neurotransmitters are produced in the pons portion of the brainstem. In particular, acetylcholine is produced in an area called the pedunculopontine nucleus. This pathway will send projections to different regions of the midbrain, as you can see here. And as well as the thalamus and the brainstem. This particular pathway is going to be really critical for regulating things like sleep-wake behavior. The second one is an area called the nucleus basalis. And this is an area called the basal forebrain. It's located at the base of the brain, and as you can kind of see, it sends a lot of different projections to the cerebral cortex. This is going to help facilitate things like learning and memory for things like riding a bike. And then additionally, we have another area in the medial septum, which is also located kind of at the base of the brain, and that facilitates memories like those that are related to events from our own lives. Now, the take-home message here is that there are multiple regions that produce acetylcholine, and depending on the areas that these regions project to, you're going to get different behavioral effects. And the more widespread these pathways are, the more variety of behavioral effects you're going to see. So let's talk a little bit about some of the different drugs that affect acetylcholine release. These primarily act on the motor neurons of the peripheral nervous system. So the first one that we're going to talk about is alpha-latroxin or latrotoxin. This is black widow spider venom. Typically, this is going to open presynaptic calcium channels, and as we are going to learn very soon, the more calcium influx that you have, the more that vesicles can dock and release more neurotransmitter. So this will produce a variety of different effects because of that enhanced acetylcholine release. You are going to get things like muscle spasms. The muscles are going to contract, and this will include rigid paralysis, including the lungs. Now, it takes several black widow bites to kill an adult. It can pretty easily kill an animal or an infant. So that's an agonist. Botulinum toxin, or Botox, actually has the opposite effect of latrotoxin. It's an antagonist, so it actually blocks release by preventing the vesicles for docking. And this causes flaccid paralysis, which means that the muscles aren't going to be able to contract. This is going to result in paralysis of the diaphragm, which means you are not going to be able to breathe. This is one of the most lethal natural toxins. It typically occurs in spoiled foods because it's bacteria. If your cans or your dairy products are bulging, get rid of them. It does have many medical uses in small quantities, including the treatment of things like migraines, as well as for cosmetic use by smoothing wrinkles and paralyzing the facial muscles so that they can't contract. I'll talk some more about this soon.